So we are now entering Unit 2 of Lab, and this chunk of Lab is going to focus on how we identify gram-positive cocci bacteria, because there are two major groups of bacteria that are gram-positive cocci that are really strong pathogens. I don't think that's the best way to phrase that, but very pathogenic organisms. And so being able to identify them is really important. So the two major genera of gram-positive cocci bacteria are going to be our staphylococci and our streptococci. So our two staphylococci species that we're going to be talking about is Staphylococcus aureus and Staphylococcus epidermidis. Now, within our streptococci, we have 5 organisms that actually belong to the genus Streptococcus, and we have 1 organism that used to belong to the Streptococcus genus, but is actually belongs to a different genus, which is Enterococcus. So we still include him in the Streptococci species that we're going to be studying. And so you can see them listed here on this screen. And as I mentioned, some of these organisms are strong pathogens. And as you can see here, Staphylococcus aureus can cause many different diseases. Like, check out that list, y'all. Staphylococcus epidermidis is much less common than Staphylococcus aureus, but can still cause disease. And it tends to be more of a hassle in nosocomial settings, which is hospital settings. And so we're going to get into the details of the diseases a little bit later, but also in lecture unit 4, you're going to learn anything you could possibly want to learn about these diseases. In the streptococci category, Streptococcus pyogenes is also associated with lots of diverse diseases. You'll notice it's linked to like impetigo, which is a skin disease. It's also associated with scarlet fever, which is more of a respiratory disease. So it can affect different parts of the body. And you can see kind of the specific diseases that each organism is linked to. I'm not going to sit here and read that for y'all, but instead, let's look at some really great, kind of terrifying photos. Okay, so otitis media is an ear infection. Pharyngitis is going to be more strep throat. Osteomyelitis, you can see the diseased state of the foot here, and endocarditis. Just going to browse through these real quick. SSS is scalded skin syndrome. Necrotizing fasciitis is going to be the flesh-eating disease, or most people refer to as flesh-eating disease. Then we have cellulitis, impetigo, which are different skin diseases, and then conjunctivitis, which is also known as pink eye. And so on the picture on the left, originally this lady had a pimple, and as you can see, the infection spread and got a lot worse. Similar on the right, what started out as an insect bite got infected, and you can see that it affected the skin quite a ways out from the initial bite. And on this one, sandpaper rash, because it looks like somebody took sandpaper to their skin, and it kind of resembles sandpaper itself. Notice that it's associated with strep throat. And so some diseases may start off in one part of the body, but then get access to the bloodstream where it can circulate and infect other areas. And one way that actually happens is through an infected tooth. It's able to get into the bloodstream and circulate. Okay, so some little tastes of some of the diseases we are going to be talking about in lecture, and we'll be touching on some of them in lab as well. But what's one thing that is important that we're going to be using throughout this unit is going to be the flow chart that is posted on Blackboard. There is a semi-filled out one. You'll want to fill out the rest of it as we go through these labs and videos. And then there's a blank one because you're going to need to know that flow chart pretty much by heart when it comes time for the lab exam. So I suggest you start on it sooner rather than later. So a little bit more information here, because this is related to diseases. When we think about Streptococcus agalactiae, and notice it says group B strep. Based on some of the biochemical properties of these organisms, we've differentiated them into different Lancefield groups, which we'll talk more about later. But you are going to need to know the group that each organism is associated with. So Streptococcus agalactiae is a group B strep. And this one in particular is important because, as you can see on the slide, it can be found in about a quarter of women in the vagina. And so while the mom may not be affected in any way, if the baby is delivered vaginally, then the baby can be exposed to the bacteria, which can have some pretty negative side effects. Okay, so if the bacteria is discovered, then antibiotics can be used either in pregnancy or labor to aid the prevention of this being passed on to the baby, and the baby can be monitored after birth if they are concerned that the baby has been infected. But it's just one of those like bacteria that may not be harmful to adults, but can be harmful to babies. And we'll learn about a couple of viruses that's the same kind of MO or same concern. But yeah, okay, so next. So we are going to talk about a lot of pneumococcal diseases, and we often refer to these pneumococcal diseases because of the bacteria species name that they're associated with, and that is Streptococcus pneumoniae. So you see the pneumo comes from pneumoniae, coccal comes from coccus. And so as you can see on this slide, there's a lot of pneumococcal diseases, right? Pneumococcal pneumonia, pneumococcal meningitis, pneumococcal sepsis, etc. Okay, but that pneumococcal is the name of the disease, and it's also giving you a hint as to which organism caused that disease. All right, so for the organisms that we're going to be studying, they are all going to be gram positive, and they are typically going to have one of two arrangements as implied by the name. Streptococci are normally going to form chains, and staphylococci are typically going to form clusters, right? Excuse me. But as you can see on the slide, if the gram stain is done from a broth, you tend to get the chains. But some species will form pairs as well. So if you see diplococci, don't be too alarmed. Just think of it as like a mini chain, okay? So before we dive into the specific tests, I also want to review the first part of this handout that's associated with this PowerPoint presentation, and kind of review some of the key terms that you're going to be hearing throughout this unit, okay? So in lab so far, we've just talked about general media, okay? Anything can grow on it. It just provides nutrients. If it contains agar, then it's a solid media, but it's general because anything can grow on it. Now we're going to start using selective and or differential media. So with selective media, only certain types of organisms can grow, others will die. But with differential media, anything can grow on it, but it will grow differently based on some of its properties, okay? So for instance, if we want to talk about salt media, which is what we used during lecture 2, I believe, salt media is going to kill most organisms because most organisms cannot handle high amounts of salt. So only halophile or haloduric organisms are able to grow. So a salt broth is selective because it's selecting for organisms that can handle high amounts of salt. Okay, so what can grow on selective media? We say that the media selects for it. So a salt broth selects for haloduric and halophilic organisms. It selects against or kills non-halophilic organisms. Okay, so on this handout, you will see just some common selective media that we are going to be talking about and what they inhibit. So this is the kind of detail you are going to want to be familiar with for your second lab exam. Now for differential media, again, that's where we're able to differentiate organisms based on their ability or inability to do something, okay? So for instance, some bacteria can lyse red blood cells completely, wipes them all out. Other bacteria can only partially lyse red blood cells, whereas other bacteria can't touch red blood cells at all. So there's kind of three different categories of bacteria based on their inability or based on their ability to lyse red blood cells. And so if you streak a bacteria on a blood agar plate, it can grow, but it may grow differently based on if it's going to be lysing some of the red blood cells in the media, if it can't lyse any at all, or if it can only lyse some. So anything can grow, but they may grow differently, okay, based on the property we're looking at. Now, some media is going to be selective only. Some media is differential only. Others are selective and differential. So as we go through all of these tests, make sure you're making note of the type of media that it is. And if it's differential, what exactly is it differentiating? If it's selective, what is it selecting for and what is it selecting against? Okay, all details you're going to need to know. So how do we know if a bacteria can do something? Can it carry out a certain chemical reaction? Can it lyse red blood cells or not? Well, there are three things that we may be looking for. One is going to be a color change. Okay, so we use pH indicators that are going to change colors based on the environment of the test tube or the plate. So for phenol broths, we're looking at fermentation. If fermentation happens, acidic byproducts are released and the overall pH in that test tube will decrease. And so the pH indicator phenol red will be bright yellow if fermentation has happened because it's an acidic pH. But if fermentation has not happened and it's more of a neutral or basic pH, then the media is a pinkish color. So just by looking at the tube, we can see a very obvious color difference. Is it yellow or is it pink? Okay, yellow means one thing, pink means another. Sometimes we'll be looking for precipitants. And so in some cases, there are chemical indicators that when they combine with the product of a reaction, it forms a precipitate. And so bile esculin agar is one of these examples. And the iron indicator that's within the agar will actually react with esculetin and form a very black precipitate. And so the tube, part of it at least, when you take it out of the incubator, will be a very dark black color, which lets you know that the reaction occurred. So again, we're looking at a color change, but instead of it being a color change due to a change in pH, it's due to a reaction occurring. And then there are other things that we can also observe. Are bubbles present? Remember we talked about hydrogen peroxide and the enzyme catalase. When catalase encounters hydrogen peroxide, it breaks it down and we see bubbles. So we know that reaction is occurring because we can visually see it. Okay, the same with the red blood cell example I gave earlier. The blood agar plate is blood red in color because it contains blood. So if a bacteria can lyse those red blood cells, you'll see the colonies on the plate, but the agar is completely clear around those colonies because the bacteria lysed all those red blood cells. Okay, But organisms that cannot lyse red blood cells, when you streak them on the blood red agar, the colonies will grow, but the agar remains blood red because the red blood cells are still intact. So you can differentiate, hey, can they lyse red blood cells? Yes or no. All right, so we're gonna stop here, okay? And the next set of videos is gonna focus on some of these specific tests. And so what you are gonna wanna pay attention to is the name of the test. Is it selective or differential, and why? Is it targeting or testing for specific organisms, and how? Then you need to know what the different results mean. If something grows, what does it mean? If it doesn't grow, what does it mean? If you see a color change or the presence of a precipitate or bubbles or whatever, what does it mean? Okay, so there's going to be quite a few tests. If you're a flashcards person, this might be a great time for your flashcards, have one per test. But as always, please let me know if you have any questions, and make sure you're keeping up with the flowchart. All right, y'all. Have a